Hi, everyone. Today we are going to finish lecture 5, the second half of it, and I'll tell you a little bit about the upcoming tests towards the end of the lesson as well. So let's recap what we've done so far on Tuesday's lesson. This is the study guide. If you have it, please take it out. If not, that's okay. You can just copy whatever I have here on a piece of paper. That works as well. So we learned about cellular respiration as a way to generate energy in our cells. And we wrote down the word equation for it, which is glucose plus oxygen producing carbon dioxide and water. And in the process, it's going to generate energy. Energy that we can capture using ADP and create ATP. The chemical equation is C6H12O6 plus 6 oxygen, and that's going to produce 6 carbon dioxide, 6 water. And for every glucose that we break down, we can generate up to 36 ATP molecules. So this is a redox reaction, which means the reactant is going to be transferring electrons from one to the other. And in the case of cellular respiration, glucose is the one that is going to be oxidized. Okay, so we're going to be removing high energy electron from it. And when we do that, it ultimately becomes carbon dioxide. And for the oxygen, it is going to be picking up the electrons, and that's going to be reduced into water. Now, this is not a direct transfer, okay? As the electrons are being removed from the glucose, they are actually captured by energy carrier, by electron carriers, which will take them to stage 4 of the reaction. And it is in stage 4 that the electrons will be used to make ATP and ultimately get transferred onto oxygen. So speaking of the stages, there are 4 stages in total. Let's just recap the first first 2 stages that we learned. Okay, we only did 2 on Tuesday, right? So the first stage is called glycolysis. Glycolysis. Glyco is sugar. Lysis is to break. Does anybody remember where does glycolysis take place in a cell?
Cytoplasm.
Excellent. That happens in the cytoplasm. Okay, so we started with 1 molecule of glucose and we generate 2 net ATP. 2 net ATP. Okay, we invested 2 and then we got 4 back. So in the end, we have 2 net ATP. In addition, we were able to remove some high energy electrons from glucose, and we will be capturing them with NAD, and that creates NADH. Okay, so if you need a reminder, do on the side here, NAD plus and FAD are electron carriers. Okay, so they capture the electron, and when they do, it's going to become NADH or FADH2. These are all in the notes we took on Tuesday. And also, we will have 2 molecules of pyruvate. Okay, 3 carbon atoms each. Started with a 6 carbon glucose. After you split it, we have 2 3 carbon pyruvate. So these ATPs that we got, they are directly produced. Okay, produced by substrate level phosphorylation. And that's what we learned last time. Let me just jog your memory a little bit. Um, there are 2 ways to produce ATP. There is the substrate level phosphorylation, which involves an enzyme transferring the phosphate from a donor onto ADP, and then you're gonna make ATP. So this is not the most common way of doing it. It's only gonna make about 11% of all the ATPs. It happens in step 1 and step 3. The other method that makes up 90% of the ATP is called oxidative phosphorylation, which we haven't learned yet. Okay, we're gonna learn that today. Right, but I just wanna remind you that the 2 ATP here that we produce in first step is produced by the substrate level phosphorylation, the enzyme helping transfer the phosphate from a donor molecule onto ADP. Okay, second step is pyruvate, pyruvate oxidation. And that is happening in the matrix. So the pyruvate that we produced from step 1 is going to enter the matrix of the mitochondria, and then it's going to undergo 3 reactions. Right? Let me go back to the note for you. It's going to undergo 3 reactions. Number 1, we're going to remove carbon dioxide. So it changes from 3 carbons to just 2 carbons in the end. And then we are going to capture some high energy electron with NAD+. And by now, you've pretty much used up the investment that we put in at the beginning, the 2 ATP that we've invested in. And so we need to recharge the molecule. Right? And that's what the coenzyme A is. Coenzyme A is going to re-energize the molecule so that it is going to react further. So over here, the key products are 2 carbon dioxide, 2 NADH, and 2 acetyl-CoA. Okay, I just want to remind you, these products are per glucose. Per glucose. And that's where we stop. Okay? That's where we stop. Any questions so far? Nope. All right. So we're gonna continue. I am going to just keep writing on this note so that when I post it, it's going to be like one continuous thing. Um, so that's number 2. Let's take a look at step number 3. Okay, so step number 3 is called the Krebs cycle, okay, which also is called the citric acid cycle. All right, so a couple of things happen here. Each pyruvate, each acetyl-CoA rather, will combine with a pre-existing. It's already there. That's what it means by pre-existing. Pre-existing 4 carbon molecule to form citric acid. Okay. So citric acid will undergo a series of reactions and release electrons. After all, we are oxidizing it, right? So then here are the key products. Okay. One cycle produces 1 ATP, 2 carbon dioxide, 3 NADH, and 1 FADH2. Okay, remember, one glucose produces 2 acetyl-CoA. Therefore, one glucose is going to have 2 cycles. So for each glucose, we're going to actually produce 2 ATP, 4 carbon dioxide, 6 NADH, and 2 FADH2. Okay, so that's all very confusing, but copy it down first. We'll draw it out on the summary chart. Hopefully it'll make more sense when we get there. Okay, just copy it down first. Okay, these are just like numbers, right? Basically, that you have to be aware of. So let's go ahead and go to the other chart here. Okay, so we already wrote down what happens with the glucose. We break it down into pyruvate. Each pyruvate will be converted into acetyl-CoA, and we actually forgot to write down the name for this process. This is pyruvate oxidation. Right? And that happens in the matrix. And now what's going to be happening is now there is going to be a pre-existing 4-carbon molecule, right? It has a name. I'm just going to write it down so it's easier to refer to it. I mean, you don't really have to know the name, but that's already there. This is called oxaloacetate. And it's going to combine with acetyl-CoA. And when that happens, 4 plus 2 is going to be 6 carbon again. And this is citric acid. Okay, so that's why the Krebs cycle is also called the citric acid cycle, because it's the first product that's formed in the cycle. And without going into too much detail, I suppose, what's happening is the citric acid is going to undergo a series of reactions. And the key thing to know here is after a series of reaction, you end up recreating the oxaloacetate, okay, which would be available to combine with another acetyl-CoA that's coming in from step 2. So it's a cycle that just keeps on going on and on and on. As long as you keep on feeding it acetyl-CoA, this is going to be able to keep going. Okay, so there are a couple of things that we can get from here. First of all, you will be producing carbon dioxide, okay, 4 of them. Um, so we had carbon dioxide here, and then we're gonna have like even more um, carbon dioxide from this step here. Actually, sorry, it's two. Um, it's four in total, two per cycle. And we will be removing high energy electron from it, which means we're going to have to capture it. So 3 NAD plus, it's going to come along and it's going to generate 3 NADH. Okay. We haven't seen the other energy carrier yet, which is the FAD. No plus for this one. And you're going to get FADH2 afterwards. Last but not least, we will also be generating ATP, but just one of them. Okay, so this is direct ATP production through substrate level phosphorylation again. So that's the key. These are the key products for one cycle. But as I have been trying to tell you, this is going to happen twice for every glucose. Right? So at the same time this is happening, the other acetyl-CoA is going to do the same thing. So you end up having 4 carbon dioxide, 6 NADH, 2 FADH2, and 2 ATP. Okay, and uh, this is the Krebs cycle. Okay. So this happens in the matrix. This is basically the summary of all the key products that you get as you break down glucose. Okay, I'll give you a moment to catch up, copy the stuff, and then we can go on from there. Okay, let me just paste this into our notes so that you will have it. Okay, so there we go. That's a summary there. Now let's go back to the study guide here for a second and fill in the third row. Okay, so the third row is Krebs cycle. Krebs is a person, by the way. And that happens in the matrix as well. Now the key products, we just wrote it down. So it is 4 carbon dioxide, 6 NADH, 2 FADH2, and 2 ATP. Once again, this is per glucose. Right? And I'm going to use the same purple star here for these 2 ATPs. Right? So 2 here and 2 here. 4 out of the 36 ATPs are produced using substrate level phosphorylation. Again, if you forget what that is, that's the one where you have the enzyme and then you have a donor molecule, and then you have ADP here. Okay, you can't see, I'm not going to write it. But basically, this one has 2P, and this one has a P, and the enzyme will put it on to make ATP again. So 4 out of 36 it's 11%, right? So I want to ask you to see if you remember what's going to happen to all these energy carriers that we produce along the way. What's their purpose? They have the energy, they have the high energy electron in them now. What are they going to do with it? Where will they be taken to?
You see?
That's right. So that's the last step. Electron transport chain, okay, or ETC for short. And this is going to be in the crystal. Okay, so all these electron carriers that we've created along the way, it's going to drop off the electron at the ETC where you will use them to produce a lot of ATP. Okay, 32 to be exact. And you will also generate oxygen. Okay, so these ATPs, they are produced by not substrate level phosphorylation, not this way, but the other way, which is called oxidative phosphorylation. Okay, that's that. So let me copy this over as well so that we have it as part of our notes. I mean, of course, like the answer keys are posted online as well, right? So you can always look at that. All right, so today is 10th. Okay, now we're gonna learn about the ETC. So that's gonna take some time, and it's probably a good idea if you start with another piece of paper.
Is it oxidative phosphor? What is
Phosphorylation,
this?
yeah. Yeah,
Oh,
phosphorylation.
phosphor.
Yeah. PHOS, PHORY, and then lation. Okay, so let me show you the note. I did it with my other class yesterday. We're gonna basically be drawing this thing, okay? So you need to start a new piece of paper and have it like sideways, okay? So draw like that, okay? So ETC happens in the crystal. So let's draw a few things here to orient ourselves. And we have the outer membrane of the mitochondria. And here is going to be the inner membrane. What is the name of the space between the outer and the inner membrane?
Remember?
Yeah, that would be the intermembrane space. Very good. And the innermost portion is the matrix. Now, these foldings are the cristae, right? And embedded within the cristae are these protein series. Okay, so there are many of them, like hundreds and thousands of them, all packed in the cristae, but we're just going to draw a few of them, and the whole group together is the electron transport chain. Okay, so the electron transport chain consists of a series of membrane proteins. And let me zoom in a little bit for you in case you're watching this on the phone. Series of membrane proteins arranged in order of increasing electronegativity. Okay, meaning attraction for electrons. Okay, just copy that down first, and then I'll explain to you what that means. Okay. Okay. So, I mean, these proteins they have their names, but for simplicity, I'm just gonna call them one, two, three. In fact, there are actually five, not three, but that's okay. So this is one, two, and three. Right? Basically, what this thing is saying is number two likes electron more than number one, and number three likes electron more than number two. That's basically what it means by in order of increasing electronegativity. So when they're arranged like this, then the following will happen. If you give an electron to the first protein, then number two is going to take it because it likes electron more. And then number three is going to take it from number two. So as a result, the electron will bounce down, will flow down. Flow down is a better word. Will flow down the ETC because of this arrangement. Okay. So that's a key thing to remember. Does anybody have any question? Okay. So it's like a game of hot potato, right? You take it and take the electron, you pass it on to the next one, and then the next one pass it on to the next one, so on and so forth, until you get to the very end of the ETC. All right. So let's take a closer look here. We have the energy carrier that was created from earlier on, right? So we have an NADH here, and it's going to drop the electron off at the first protein. And after NADH drops off the electron, it will become NAD plus again. Okay, and then the NAD plus will go pick up more electron. Right, it'll go back to the Krebs cycle and pick up more electron. It'll go back to pyruvate oxidation and pick up more electron, right? So that they could bring it to the electron transport chain. So there's the electron that got dropped off. Right? It's not just one, but I'm not going to draw too many of them and create confusion. So as I was telling you earlier, once the electron gets on the ETC, it's going to get passed down the proteins. So number one will pass it to number two, which will pass it to number three. Okay, so I'm gonna write the note here. Um, let's use black here. NADH and FADH2 drop off electron at the ETC. As electron flows down the ETC, its energy, the electron's energy, is used to pump H plus from the matrix into the intermembrane space. Right. So let me explain first, and then I'll give you some time to copy. Basically, when the electron is flowing down the ETC, we use the electron's energy to pump H plus. Okay, pump H plus from the matrix to the intermembrane space. Okay, every time the electron flows down the ETC, you will keep on pumping H plus into the intermembrane space. So over time, as this continues, you will have lots and lots of H plus in the intermembrane space. Okay, so this is going to be high concentration of H plus, but low concentration here. Okay, there will be some H plus, okay, but we pumped a lot over. All right, so that's what it means. NADH and FADH2 will, I'll draw the FADH later, okay? NADH and FADH2 drop off electrons at the ETC, and the flow of electron creates power energy that will allow us to move H plus from matrix to intermembrane space. Right? Now, this process of pumping them over, pumping the H plus over, requires energy because we are doing active transport. As you may recall, active transport is transporting things from low to high concentration, which is what's happening here. Low concentration of H plus in the matrix and high concentration, excuse me, in the intermembrane space. So you keep on pumping more and more over. Okay, copy first. Let me know if you have questions, and then I'll explain more.
It's like protein pump against concentration gradient.
Exactly, right? So this would be an example of protein pump like the active transport note that we took, right? Okay, I'm just waiting for you guys to copy. If you're done, give me a signal, and then I'll keep talking. Now, while we wait for you to copy that, let me just also tell you about some of the stuff. So I got a few emails from people about the last quiz that they're saying, it's a little bit stressful because not enough time. So for the test next week, I will give you more time, okay? I'll give you more time. Um, the test is going to be on Thursday. We'll have a review on Tuesday. You should start reviewing on your own this weekend, of course. And the test will be lecture 1 to 5. All this will be posted online later, okay? So like if you don't have to write down. Lecture 1 to 5 plus the lab. That's gonna be the test material. And it is going to have, let me see, 44 multiple choice and 10 multi-select questions. Okay, so 44 regular multiple choice question, 10 multi-select questions. Those are the one that you guys don't like, right? With more than one correct answers. And there will be no short answer this time. No short answer. Um, there is going to be a practice review test that I'm going to post for you, and you can try that out. Okay. And the time limit, did I tell you time limit already? It's going to be 100 minutes. Okay. 100 minutes. There's a question. Are we going to redo the lab work? No, no. Okay. The lab is done. Okay. Any question about the test? Here, I'll post the announcement later on. This is for my other class, but this is the same as you guys, okay? 44 multiple choice, 10 multi-select, no short answer. You don't have to type anything. 100 minutes should be enough. Um, and then there's going to be a review test for you to try, but you will only be able to do it 5 times, okay? Because I don't want people to keep on doing it and wasting time. You just do it like 2 times max. But I give you 5 times. Any questions at all? Comments as well. If you want to say anything about the quiz, feel free to do it. Feel free to tell me. I'm not
I
gonna
have
take
a question about the lab part of the
sure.
test.
Yeah, it's
So,
just a few questions. I think it's four or five questions. We'll take up the important aspect of the lab um, on the review on Tuesday, so you know the right answer.
okay,
Yeah,
thank you.
yeah, because you can say, how do we study for it if we don't get the lab back, right? I assume that was uh
Yeah,
yeah, yeah,
exactly. Yeah.
right. Yeah. So I will post the review for the test. You will see it today, and then we'll take it up. Okay. So everything you need to know regarding those lab questions, we'll go through on Tuesday. Anything else? Okay. Well, if you have anything at all, feel free to email me. Okay. If you have any concerns or suggestions, or even if you think something is not too good, right? Like, oh, maybe you didn't have enough time or whatever, just let me know. Okay, I'll take those things into consideration. Um,
Metabolism is also included in the
yes,
test.
metabolism is also on the test. It's
Oh,
everything that we learn up to the point of this point of the course. Okay, one, two, three, four, five, and the lab. Okay, so NADH and FADH2 drop off. We talk about this and how the electron is gonna help pump the H plus over, right? Now I'm gonna you know use an analogy to help you understand the situation here. It's up to you if you want to draw this or not. You can just listen, okay? So let's say there is a waterfall, okay? Maybe don't draw, okay? Maybe just watch. So then the water is gonna fall down, okay, like this, right? Okay, that's the waterfall. Okay? So that's a lot of energy as the water moves over the edge, and it's generating a lot of energy because of the movement of the water, right? So people have invented a way to harness that energy by building a dam. Okay, so this is a dam. Okay, so by building a dam, what happens is you block the flow of water, and then the water is like all built up here. Okay, all built up. And then what we can do is to create an alternate path for the water to flow through. And along the way, we're going to put a turbine in there. Okay? So when you open the gate and let the water go through, okay, so water is going to be rushing through here. It's going to turn the turbine and then it's going to flow down like that, right? Okay, there's water coming. The water will naturally go towards a lower spot. Okay, so this is going to turn the turbine, and that's how we create electricity using, hydropower. I mean, that's not the only way to create electricity, but that's one way of doing so. All right. So the situation is similar here. Instead of water, we basically have a bunch of H plus that we have built up in the intermembrane space. Okay, so this is intermembrane space. Okay, they want to go back into the matrix. Okay, because things naturally wants to go from high concentration to low concentration, but they cannot do so on their own. The dam is blocking it. So in our case here, the dam is like the inner membrane. The inner membrane is the same kind of material as the cell membrane. It does not allow charged particle to go through. So even though all these H plus is very high highly concentrated here and they want to come back in, the membrane blocks it. It dams it. Okay? Is that okay? You guys understand? So the only way to come back in is to go through the turbine. And there is a special protein that is in the cristae. Okay? And it looks like a doorknob. It looks like the turbine that we drew. This is called ATPase, or sometimes called ATP synthase. Okay, depending on what you read. So these H plus ions, they will flow through the turbine and go back into the matrix. And as they are doing so, as they are passing through the turbine, this is where it's going to take ADP and P and combine it to create ATP. Okay, I'll write down for you as H plus moves through ATPase, ATP are generated. This method of ATP generation, ATP production, is called oxidative phosphorylation. That's it. That's oxidative phosphorylation. 32 out of the 36 ATPs are produced this way. Okay, 90% are produced this way. So I have two more things to add to the diagram. Seriously, any questions? Well, the good news is after test one, you won't have to worry about these materials anymore, right? Because the second half is standalone compared to the first half, right? Okay. You won't be tested on these things anymore on the final test. Okay. Does anyone remember from last class how many ATPs can you produce from one NADH? Let me just write it here for you. One NADH is equivalent to how many ATP? Does anybody remember? Will we
Three.
be revisiting these topics on the next semester though?
Two.
No, next semester is all about human bodies. Yeah. I mean, there will be some connection, but not like, direct connection. Sorry, I interrupted. Someone was talking. How many? 3, right? Yeah. Okay, very good. And one FADH2 is equivalent to how many ATP?
Two.
2, yeah. Okay. All right. Now, this is an oversimplification, but you can think of it like this. The NADH is drop off the electron, and then you're going to pump like 3H plus over, and then when these H plus come back in, 3 ATP, right? Now, FADH2 has less energy compared to NADH because of the 3 and the 2 rule that I told you about. So it's not going to be able to drop off the H plus in the first protein of the ETC. Instead, it's going to drop it off at a later point. Okay? Now let me zoom in a little bit So when it does that, the electron will have less time to provide the energy for pumping H plus. So instead of doing 3 times, we're just going to be able to do it 2 times. Okay, it's the same hydrogen as the other ones. Okay, I'm just using blue here to kind of make a distinction. Okay, so you have a lot of H plus here. And so when these guys come back in through the doorknob, the turbine, sorry, the turbine right here, it will only be producing 2 ATP. Nevertheless, it's still ATP, right? Just find the right shade again. It's the same thing. ADP plus P becoming ATP. All right? So that's that. And of course, the FAD is going to go back and pick up more electrons, right? So this is what happens in the ETC. All right. Just one final thing, and then we're done with the note for now. I'll give you another analogy to help you understand what's going on, okay? I remember when my kids were younger and I took them to the park. This is a very familiar uh, situation, okay, scenario. Okay, so like they go on the slide, and I always remind them like don't go down the slide unless it's clear. Okay, but every now and then there is a kid that's like at the bottom after they go down the slide, they stay there. Okay, they're for some reason they don't want to get off. Okay, they just lie down there, right? And then it kind of blocks the whole slide. Like the next person cannot go down. They want to kick them in the head or something like that. And so the parent have to come along, right? And then get the kid off the slide so that the next person can go, right? So remember this analogy when we talk about the ETC. The slide is the ETC, and the electrons are the kids, right? Trying to go down the slide. So when the electron goes down the ETC at the very end, it doesn't know how to get off on its own. Okay, it needs a parent to pull it off, so to speak. Okay, and we need to pull the electron off at the end because otherwise the next electron is not going to go. Okay, so that would be the job of oxygen. It's getting really, really messy here. But let me just try to find a spot for it. Here is oxygen. Okay, oxygen is going to pull the electron off, and then it's going to become water. Okay, oxygen is going to pull the electron off, become water. Okay, so I'm going to write a quick note here. O2 is it's called a final electron acceptor. That pulls electron off the ETC. This allows more electron to flow down the ETC. And that's why we have to breathe. If we don't have oxygen in our system, then there's nothing to pull the electron off. Okay, and then the ETC gets clogged up. The electrons can't flow anymore. Okay, try to listen for a second, okay? Without oxygen, there will be no electron flow. Without electron flow, we cannot create the H plus here. It's like we cannot dam the water, right? So if we don't build up this H plus concentration, then they won't flow through the turbine, and we won't be able to produce ATP. Okay, everything that is important that happens in the body needs ATP, right? Beating of the heart, functioning of the brain, all these things needs ATP. So without oxygen, We can't do any of those things because we're not generating ATP, and then we die. So that's basically the whole story. Are we okay? Let me know, please. It's really hard to gauge whether you guys are with me or not. Yes, go ahead. I'm repeat.
What is the direction of the electron flow?
We'll just say down the ETC, okay? Because each subsequent protein has a higher attraction for the electron, okay? So as soon as you give it to number one, number two is going to pull it off number one, and then number three is going to pull it off number two. And as such, it creates a flow of electron. Okay? But ultimately, oxygen has to come. Oxygen has the highest electronegativity compared to all the proteins. So that's why it's able to pull the electron off the chain such that the next electron can slide down. Hope that answers your question. And I mean, like, it's not something that, you will necessarily fully grasp the first time you hear it, but that's what the recordings are for, right? And please feel free to send me email if you guys need any clarification. Okay. So I'm going to go back to the study guide here for a second, and I will copy this table over, and we'll fill it in together. Okay, this table is about trying to keep track of all 36 ATP produced in the process. All right, I'm going to put it right here. So if you have the study guide, please follow along. If you don't, just copy it down, I guess. It's not that difficult to reproduce this chart. Okay. 4 stages, right? Okay, we have 3 stages here. We skip the ETC here. You'll see why in a second. Actually, ETC is right here, right? So let's keep track of the ATP. How many ATPs are produced through substrate level phosphorylation? That's the one where the enzyme comes and transfer the phosphate, right? How many net ATP do we produce using this method in glycolysis? That's right, Cheyenne. It's 2. So 2 ATP, 2 net. And what about energy carrier? How many energy carriers were produced in glycolysis?
4.
Sorry, how many?
4
Um,
energy carrier produce.
is it 4 though? Like, let's take a closer look at the summary chart or the flow chart that we drew. How many NADH do we produce?
3.
Okay, let's take a closer look here. I guess we can just scroll up here, right? That's the glucose, and we split it into the pyruvate, and there is the energy carrier, right? NADH. Just 2, right, guys?
Yeah,
Just 2.
2.
Yeah, just 2. Okay. So, like I said, if you have trouble memorizing these numbers, which is totally understandable, please have some help sheet next to you when you do the test. So that's going to be 2 NADH. Now, there is a little bit of a problem here to make things slightly more complicated. Where are these NADH produced? Okay, this is glycolysis. So where are the NADH produced? Like which part of the cell are they produced in?
Cytoplasm.
They are produced in the cytoplasm. And the the purpose of these energy carrier, the NADH and FHDH2, is to transport the electron to the ETC, right? And which part of the cell is the ETC located at?
The crystal.
Exactly. Okay, so now bear with me for a second, please. Okay. I mean, you don't really have to draw this. You can just watch and listen. Okay. This is the outer membrane. This is the inner membrane. This is the ETC. Okay. So glycolysis happens here, which you already know. Okay. So we've created two NADH here, and it's supposed to take it to the ETC. But the problem is NADH is actually quite big, and it's not able to cross two layers of membrane by itself. Okay, so what it's gonna do is it's gonna drive the electron all the way up to the border, and they're like, hey, this is as far as I can take you guys. Okay, so please get off and cross the border yourself. And then it will become NAD plus again. Okay, because the molecule is too big to cross. And then the electron will cross themselves. And on the other side, in the matrix, something else will come pick them up. Okay, the FAD will come, and then they'll become FADH2. So there is a bit of an extra step here to consider. And you might wonder, hey, how come it is FAD that's picking them up? How come it's not like, we started with NAD? Shouldn't it be the NAD that picks them up? Well, as the electron cross the membranes, the outer membrane and inner membrane, it kind of used up some energy, right? So instead of NADH, now we have FADH2. Essentially, for the crossing, you have to pay 1 ATP to do it. That's like the simplified version, okay? So in our table here, the 2 NADH, which again are produced in the cytoplasm, they will become, well, I mean, they themselves don't become the FADH2, but they will pass the electron onto the FADH2, which is in the matrix. Okay, and then the FADH2 will take the electron to the ETC the rest of the way. Now, stay with me, please, please, please. Once it gets to the ETC, then it's this stuff that we discuss. Right? It's gonna, flow down the ATC. We're gonna pump the H plus over, okay, creating a dam. When they come back in, that's how we get ATP. So tell me, how many ATPs are produced at the ETC? How many ATP are produced at the electron transport chain? 4, exactly. Very good. Okay, it's a private message that I received, okay, in case you think I'm, again, talking to myself. So we're going to times 2 here. 2 times 2 is 4. Okay, so ATP produced at the ETC, I should add by the process of oxidative phosphorylation. Yeah, again, phosphorylation is a big word, but all that means is adding phosphate onto something. In this case, we are adding the phosphate to ADP, diphosphate, to create ATP. Okay, and this is powered by oxidation reaction. Because where did you get those electrons? Where did you get those high energy electrons? We got them by oxidizing glucose, right? So that's why it's oxidative phosphorylation. In any case, if you add up all the ATP, the ones that are produced directly through substrate level phosphorylation, the two, and those that are produced indirectly, glycolysis is responsible for creating 6 ATP in total. 4 plus 2 is 6. Okay. Now, let's try this next step. How many ATPs are produced directly, okay, directly in pyruvate oxidation? Just look at the chart. That's right. Uthman said 0. Do you guys agree it's 0?
Is it
I-
1?
Uh, it's 0 actually. Let's go back here and take a look. We are doing pyruvate oxidation, right? So look at all these things, right? No ATP at all, right? Does that make sense?
Yes.
Okay. What about energy carrier? How many energy carriers do we produce in stage number 2? This is stage 1, this is stage 2. Again, just look at the chart, look at the table. How many NADH do we produce?
Is it 3?
And where do you get the number 3? I'm just curious. So that I can help you find the right answer, right? Yeah.
Because in the notes, it says the key product is 1 cycle produce 1 ATP, 2 carbon, 3 NADH, and 1 FADH.
Oh, you're looking at Krebs cycle. That's why. Okay. We're still at stage number 2, which is the pyruvate oxidation. Does that make sense?
Yes.
Okay. Very good. So it's 2, right, guys? 2? 2 NADH. And where are these NADH produced at? Where in the cell are they produced at?
Mitochondrial matrix.
Very good. It's already in the matrix, right? So we don't have to worry about this, transfer of electron. They're already at the matrix, which means you could take them directly to the ETC. So this time, when it gets to the ETC, what number do I multiply by to figure out the number of ATP?
0.
Multiply by 0?
The 1?
Okay.
No
1.
ATP here.
Is it by 2?
Is it by 2? Okay. Anyone
Yeah.
else?
No
All right.
ATP.
This is the important part, guys. We wrote it down last time, so let's go back here to revisit. All right. I guess it's important to know where to look for information in the notes. Is this thing that I highlighted last time? Okay. Let me triple highlight it. Okay. This is important because it will help you answer a lot of questions. Okay. So you should remind yourself 1 NADH is equivalent to 3 ATP. And then 1 FADH2 is equivalent to 2 ATP. Okay. They are like currency. Okay. Like what is it worth? Right? It's like saying, a loonie is $1, a toonie is $2. So an NADH is like 3 ATP, $3. Okay. 1 FADH2 is 2 ATP. Okay. This part is what you need to answer some of the questions on the test. Okay. So let's go back here. We have 2 NADH. How many ATP can they generate each?
3.
3. So 2 times 3 is 6. Okay. If you don't understand where the numbers came from, please ask me. I will show you how to find the numbers. Okay, because you need to know where to look for the numbers, even though you have your notes with you on the test, right? Like it's not going to be helpful if you don't know what to look for, right? Are we okay about where the 2 and the 3 and the 6 came from?
Yeah.
Thank you. The rest of the class good? Okay. All right, so let's add up these things. 6 plus 0 is 6. Okay. I'm gonna give you 5 minutes, and we can go on a break or whatever. Okay, but like use this time during the break as well to try to fill in the last row here. Okay, you have all the numbers. We wrote it down several times already. You just have to put them down here and do the right math and then figure out the number. Okay, I'll take it up with you when we come back in 5 minutes. See ya. Okay, we are back. Let's hear it. Does anybody have some numbers for me? Number of ATP produced directly. I'm
Two.
sorry, what was it?
280.
2, that's right. Very good. Okay. And then number of energy carrier. There are a bunch here. Let's see what we have.
6.
Yeah, okay, 6. Which one is it?
180. 180H.
Good. And
And 2 FADH2.
very good. Okay, I hope the rest of you also got this. Again, it's like all summarized here in the chart earlier, right? Now, these are already in the matrix, of course. So we don't have to transfer them. All we need to do is take them to the ETC. And what do we multiply the 6 by?
3.
3, because each NADH can produce 3 ATP. 6 times 3 gives you 18. Times 2 here, which gives you 4, right? So 18, oops. 18 plus 4 plus 2, 24. And you add up these things together, 6 plus 6 plus 24. That's how you get the 36 ATP. Okay? All right. Let's do some poll questions. Actually, you know what? I'm gonna post the answer for the poll questions for you, but let's do one here. This is a good one. Give me a second, guys. Oops, what is going on? Okay, that's the question. I'll post it here. And let me do the poll. Okay, let me just clarify. I'm not just looking for the 2 ATP, okay? I'm looking for like all the energy carriers as well when they go to the ETC, so collectively, if you do the Krebs cycle 5 times, how many ATPs are produced? Okay, let's take a closer look at this. All right, so this is 5 turns of the Krebs cycle, okay? Let's look at 1 turn of the Krebs cycle. Okay, what are being produced in 1 turn? Let's go back here. Okay, 1 turn is 1 cycle, right? So it is 3 NADH, 1 FADH2, and 1 ATP. Okay, let me go back down here. 3 NADH, 1 FADH2, and 1 ATP. So this we're going to times 3, which gives us 9, times 2, which gives us 2, and this is just 1. So if you add them up, it's 12 ATP per turn. Okay, times 5, you get 60. I know why everybody chose 120, because you are thinking about per glucose. Each glucose will turn twice, but this is not talking about 5 glucoses, right? It's talking about 5 turns of the Krebs cycle. Does that make sense? Do you understand why there's a difference? Okay. Five turn is like doing this 5 times, not like, twice. Uh, that would be like 10 times, right? You know what I mean? Okay. So watch out for that on the test when you're doing it. Okay. Have a calculator next to you as well. These number drives you crazy. It can't be worse than math, right? This is very little math. We actually will be doing more math in the second semester. Sorry, second half of the course when we do genetics. Okay. But like I said, right? Like I strongly encourage you guys to keep this with you on the test. It's totally fine. It's a reference sheet. I don't expect you guys to memorize all these things. My in-person class, they have to memorize it, okay? But like you guys have the advantage of having these next to you, okay? So take advantage of that. All right. We are going to finish the PowerPoint slides. All the hard stuff are done, okay? All the hard stuff is finished. We just have to do a few other things and then we're going to be done. Now, of course, like read through the PowerPoints if you need to get more like details from it. But I think I cover everything in the notes. Okay, so next we're going to talk about disruption of oxidative phosphorylation. There are various poisons that could interfere with the production of ATP by blocking the ETC, inhibiting the turbine, the ATP synthase, or making the inner membrane leaky. So let's take a closer look at each one of these. First is rotenone, cyanide, and carbon monoxide. Okay, you probably heard of the last two and not so much the first one. The first one is used as an insecticide and pesticide, okay, to kill insects and pests, right? And how it does that is it binds to the first protein in the ETC and blocks it. Remember, the ETC has 3 proteins. So if the rotenone comes and block the first one and make it not work, then the NADH will not be able to drop off the electrons, right? And if you don't drop off the electron, you cannot produce ATP. Is this going to affect FADH2? Is this going to affect FADH2? FADH2, as you may recall, drops it up at the second ETC protein, right? So blocking the first one is not going to affect it. But it's not really helpful because, we only create 2 FADH2, right? Which is only equivalent to 4 ATP. So if you have this stuff in your system, it's still going to be pretty toxic. Okay. Carbon monoxide. Carbon monoxide is tasteless, colorless, odorless. You can't see it, you cannot detect it. That's why you should have a carbon monoxide detector at home, right? And if you have one, where should you put it? Do you guys have a monoxide detector at home, carbon monoxide? I think by law you need to have one. But where are you supposed to keep them? Do you know? Not to be confused with a smoke alarm. Okay, close to the ground. Someone say. Well, you should keep it where you sleep, okay? Because that's the most likely place where you're gonna be poisoned by carbon monoxide and not know about it. Okay. Like why are you sleeping, right? So that's why you need to put it there. We'll learn more about carbon monoxide poisoning next semester. But basically what it does is it binds to hemoglobin, which is the protein that transport oxygen for you. Um, and so instead of transporting oxygen, it's just like transporting carbon monoxide, which you cannot use. Okay, remember, we need the oxygen as a final electron acceptor to remove the electron from the chain so that the flow of electron can continue, right? Same thing with cyanide. Okay, cyanide in chemistry class, you probably learned about cyanide, right? The chemical formula for it, it's CN. The poisonous form of cyanide, it's usually like hydrogen cyanide, okay? And it also binds to the ETC and blocks it. Okay, so basically all these things blocks the ETC, no electron flow, no H plus dam, and no ATP production. There is the oligomycin. I don't have a slide for that, but it inhibits the ATPase. Basically, inhibit the turbine, right? So that's equally bad. The H plus cannot flow through. No ATP, dead. Okay, there's something called DNP. DNP is something that was originally used as like a photographic chemical, as well as, in manufacturing fertilizer and dye and that kind of stuff. It's not meant for consumption, all right? But there was an accidental ingestion by one of the workers who worked at these plants, and he reported rapid weight loss afterwards. Okay. So when the pharmaceutical company caught wind of this, they thought they hit the jackpot with diet pills, okay, with weight loss pills. So they created this pharmaceutical version of DMP and then entered the market around 1934, okay, which was like a long time ago, okay? Over 90 years ago, if you do the math. So what happens is people actually was able to lose weight from this, but then people also started dying. Okay, and the reason is because the DMP they didn't fully understand how it worked. They just know that it kind of worked, and then later they found out that DMP actually caused the hydrogen to leak through the inner membrane space. Okay, so in this picture you can see supposedly we have all these H plus that we pump into the intermembrane space, and they were not able to come back in. They are only supposed to go through the ATP synthase. But in the presence of DNP, they are able to leak through. Okay. So instead of going through the turbine, they just leak all the way back into the matrix on their own without generating any ATP. Right? So your body thinks you're starving because you're not creating enough energy. And that's why it switched to burning the fat. Okay. And that's how it results in rapid weight loss. Right? But there was a lot of side effects associated with this, right? You know, heart problems, endocrine problems, and people die from these things. So this is no longer a weight loss medication. Okay, done. Counting ATP, we did that already. Now this slide talks about how even though we as in like our cells prefer to use glucose as the main source of energy. Okay, the cellular respiration equation is glucose plus oxygen, right? Producing carbon dioxide, water, and energy. But as you know from our diet, we don't just eat carbs, right? In fact, if you only eat carbs, it's not a very balanced, healthy diet. You're gonna have some health problems. So the proteins and the fats, all these other macromolecules that we consume can actually be fed into cellular respiration at different stages. For example, fats, we learned in lecture 2, it's made up of glycerol and fatty acids. And the glycerol can be used in glycolysis, whereas the fatty acids can be used and be converted into acetyl-CoA. Okay, and from acetyl-CoA, it goes into the Krebs cycle. Proteins, of course, are made up of different amino acids, 20 different kinds. And depending on which amino acid it is, some of it can be used in glycolysis, some of it can be converted into acetyl-CoA, some of it can be used in the Krebs cycle. Okay, so the point is, you might not be able to generate like 36 ATP all the time from all these other molecules, but you will still be able to generate ATP depending on where you feed it into the process. All right. Okay, let's take a look at this question, and I'll go through it with you. Hydrogen cyanide (HCN) is poisonous I told you this already. It binds to one of the membrane proteins in the electron transport chain and blocks the movement of electrons. If cyanide poisoning occurs, what would happen to the pH of the intermembrane space? Okay, so hint, low pH is acidic, okay? Low pH is caused by more H+. So let's take a closer look. You should be familiar with this drawing by now because we drew it together. And there is the ETC. This is the intermembrane space, right? So normally what happens is we pump H plus into the intermembrane space. That's the dam I have been talking about, right? You keep on pumping the H plus over. That's what happens normally. In cyanide poisoning, this is not happening because we block the ETC. So we have less H plus, okay? Less H plus. Less H plus equals to higher pH. That's the answer. Okay. Again, the whole point of this question is to see if you know what happens to the H plus. If you block the ETC, you cannot harness the energy from the electron. You're not pumping the H plus. Less H plus in the intermembrane space, higher pH. Okay, we're just gonna wrap up the lesson with fermentation, right, which is anaerobic respiration. So fermentation is an important industrial process for making various kind of wine or beer, alcoholic beverages. And if you bake at home and you use yeast, that fermentation process is also important for causing your dough to rise, to leaven. So humans are, our cells are able to do fermentation as well. Okay? So in muscle cells, when the oxygen demand is greater than oxygen supply, then the cell will switch to fermentation. The kind of fermentation that we do is called lactic acid. Lactic acid fermentation. We don't do it all the time. It's not sustainable with life. In fact, if you just rely on it, we only do it when our muscles are not getting enough oxygen. Okay? Like you're running at full speed for 3 minutes, and then your muscle is demanding too much oxygen. You're not going to be able to supply it. Okay, but you can still go on for like another 10 seconds or something before it completely give up. Okay, so during that 10 seconds of extended duration, your body basically switches to fermentation in those muscle cells. Okay, so what is the purpose of fermentation? Okay. The purpose is to provide an alternate path for dropping off electrons. Normally, you have to drop the electron at the ETC. And if you remember about the slide, right, and the kids, when the electrons slide down the ETC, oxygen has to be there at the bottom to take away the electron before the next electron can go down, right? But we have a problem here. We don't have enough oxygen during strenuous exercise. Okay, so the oxygen is not available to pull away the electron, which means the ETC is clogged up. There's no flow. Okay, so all these NADH has nowhere to put down the electron, and that's why the body switched to fermentation. Okay, it allows the NADH to drop off the electron elsewhere so that we can allow glycolysis to keep on happening. We wrote this down actually, if you have the note from Tuesday. The first thing I wrote down for glycolysis is that it is anaerobic. You do not need oxygen for glycolysis to take place. Okay? So glucose becomes 2-pyruvate, that's glycolysis. Right now, we don't have enough oxygen, so we cannot drop off the electron at the ETC. Instead, the NADH will give the electron to pyruvate. And in doing so, like I'm skipping a few steps here in between, but in doing so, the pyruvate will become lactate. Okay? Lactate is same as lactic acid, okay? For us, there is no difference. And then you basically empty the taxi, you empty the NAD plus, and it's going to be able to go back to glycolysis and allow it to keep on going. Right? So in this process, you will still be generating 2 ATP. It's a far cry from 36 ATP, but it's better than nothing. It will keep you going for that extra 10, 15 seconds. Okay? If running is like a difficult thing for you to visualize, just think about like hanging onto a monkey bar, okay? You've hung on for as long as you can. You feel the muscle burning. The burning sensation is actually the lactic acid, okay? And your body's about to give up, okay? But you can still hang on extra, 5, 10 seconds. That extra 5, 10 seconds is powered by lactic acid fermentation. Okay. And that's why you cannot go on any further. Because 2 ATP, while it's something, is not enough for, continuous continuously sustaining the motion any further. So this is what it looks like. We have the glucose, which is 6 carbon, and we get the 3 carbon pyruvate. Pyruvic acid, same thing. And the NADH is going to give the electron to pyruvate, and we generate lactic acid. Okay. So if you have a good workout, the next day you feel sore and the burn, partly it's because of lactic acid that builds up. Now, in other microorganisms like yeast and some bacteria, they don't have the mitochondria in them. Okay, they cannot do the Krebs cycle or pyruvate oxidation. They lack these machineries to do it, and they rely solely on fermentation. The kind of fermentation that yeast perform is called alcoholic fermentation. Okay, because it generates ethanol. Ethanol is drinking alcohol. Okay, not every kind of alcohol can be consumed, right? Sorry. Like rubbing alcohol, it's like poisonous. You can't drink that stuff, right? Okay. So ethanol is a drinkable alcohol, and then that's the product of alcoholic fermentation. And it also produces carbon dioxide in the process, which caused the bread to rise if you use yeast for baking. Now, that's what the yeast look like, and it's the same kind of concept, right? They do glycolysis, but since they don't have the ETC to drop off the electrons, the pyruvate will just pick up the electron and in the process generates ethanol and carbon dioxide. That's the process. You can look at that yourself. And basically, there are 3 possible pathways at the end of glycolysis. Right? For humans, most of the time, after glycolysis, it's going to go and do pyruvate oxidation, Krebs cycle, and ETC. Right? That's when O2 is available. Okay, that's aerobic oxidation. And if it's not available, if there's not enough oxygen, then you will do lactic acid fermentation. Okay? If we are talking about something like a yeast or other simpler organism, then they will do alcoholic fermentation. We do not do alcoholic fermentation. Alcohol is toxic to our cells. Okay? So that's only for yeast and other kinds of simpler organisms. And there you have it. This is the first half of the course, guys. You survived. Most of it anyways. Look, I guess after the test, you will officially survive the first half. All right. It's a lot of material. I understand that. I will post a review for you to to start preparing for the test. Like I said, there will be a review test that you can do as well. But Tuesday we will be taking up the review. And I mean, if you have any questions specifically, like things that are particularly confusing for you, email me and I'll make sure I cover it on the review as well. Okay, thank you guys. See you and have a good long weekend. Okay, try to get some rest, even though there are a bunch of tests coming up. See you Tuesday. Bye bye.
Hey, Prof.
Yeah, go ahead.
So I'll be getting a laptop today.